At 11,000 meters below the surface, steel implodes. Glass shatters. Human tissue collapses instantly. Yet here, proteins remain folded. Cells remain alive. Life continues. Pressure at this depth exceeds 1,100 atmospheres. That is not force. That is molecular violence. Pressure doesn't break bones first. It attacks proteins. It distorts their shape and destroys function at the atomic level. Enzymes misfold. Membranes solidify. Electrical signaling collapses. By every known rule of biochemistry, life should end here. But it doesn't. Deep-sea organisms do not resist pressure. They neutralize it. Inside their cells exist molecules that do not strengthen structure. They rewrite physics locally. These molecules are called piezolytes. The most powerful among them, TMAO, trimethylamine N oxide, does something extraordinary. It surrounds proteins and stabilizes hydrogen bonding under compression that would normally tear structures apart. Pressure squeezes. TMAO counter-squeezes. The result? Proteins behave as if the pressure isn't there. Membranes don't harden. Lipid chains remain flexible because their saturation is evolutionarily tuned atom by atom. Even ion channels, the electrical gates of neurons, remain operational in darkness and crushing force. These adaptations are not rare. They evolved independently in fish, crustaceans, mollusks, and microbes. Evolution didn't solve pressure once. It solved it again and again. If proteins can be engineered by nature to function under crushing force, then the limits of life are not where we thought they were. Not on Earth, and maybe not beyond it. The deepest ocean is not a graveyard. It is a laboratory where biology learned how to survive the impossible. At extreme depth, pressure doesn't just threaten life. It should freeze it. Cell membranes, the thin, fragile boundaries that keep cells alive, should turn rigid, brittle, useless. And yet, in the deepest ocean on Earth, cells remain soft, flexible, and functional. A cell membrane is not a wall. It is a living fluid. Under crushing pressure and near freezing temperatures, that fluid should solidify. Transport should stop. Signals should fail. Cells should suffocate from the inside. By textbook physics, deep-sea life should shut down at the membrane level alone. But evolution rewrote the membrane itself. Deep-sea organisms don't reinforce their cell walls. They re-engineer their lipids. Fatty acid chains are lengthened or shortened. Unsaturated bonds are positioned with atomic precision, and membrane composition is tuned molecule by molecule to remain fluid under immense compression. While pressure tries to lock lipid chains together, these membranes keep moving. They bend, they flow, they breathe. Ion channels continue opening and closing. Nutrients still pass through. Electrical signals still travel. Not despite the pressure, because the membrane is built for it. This solution didn't evolve once. It appeared independently in deep-sea fish, crustaceans, mollusks, and microbes. Different lineages, different chemistry, same physical outcome. Evolution didn't fight pressure. It learned how to live as if it weren't there. If living membranes can remain functional under forces that should freeze them solid, then the basic requirements for life are far broader than we imagined. Pressure is no longer a boundary. It's just another variable biology can tune. In the deep ocean, life doesn't harden. It stays soft and survives because of it.
At extreme depth, pressure does not merely squeeze matter. It reshapes the energetic landscape in which chemistry occurs. Every biochemical reaction depends on fleeting molecular motion, enzymes flexing, substrates aligning, transition states forming for fractions of a second. Pressure resists all of this by reducing available volume and penalizing any reaction step that requires structural expansion. Under these conditions, classical reaction theory predicts dramatic slowing because activation volumes become energetically expensive. In most surface organisms, even modest pressure disrupts catalytic timing long before any visible damage appears. Deep-sea organisms avoid this trap by altering something subtle but decisive, the internal compressibility of their enzymes. Their catalytic proteins are built with microcavities, flexible loops, and hydration shells that redistribute pressure at the angstrom scale. Instead of collapsing, internal motions are redirected, allowing catalytic cycles to proceed with near-normal kinetics. At the same time, intracellular water behaves differently. Solutes, such as trimethylamine N oxide, reorganize hydrogen bond networks, reducing pressure-induced viscosity and preserving diffusion rates that would otherwise grind metabolism to a halt. The result is not faster chemistry, but chemistry whose energy barriers barely change even as external pressure climbs beyond 1,000 atmospheres. From the outside, everything should be slowing. From the inside, reactions remain synchronized, energy continues to flow, and time, at the molecular level, keeps its rhythm. Far below the surface, survival is not about resisting force. It is about redesigning motion itself. At extreme depth, pressure does something far stranger than crushing matter. It bends time at the molecular scale. Chemical reactions are not governed only by structure. They are governed by speed, by how often molecules collide, how fast bonds rearrange, how quickly energy can cross microscopic barriers. Under immense compression, those rules shift. Diffusion slows as free volume disappears. Substrates reach enzymes less frequently. Activation energies rise, not because chemistry changes, but because motion itself becomes expensive. An enzyme can be perfectly folded and still fail, not from damage, but from delay. Deep-sea cells compensate by tuning reaction networks rather than single molecules. They favor pathways with fewer intermediates. They bias metabolism toward reactions with lower entropic cost. They trade efficiency for reliability, accepting slower yields to preserve continuity. Even ion flow changes character. Proton gradients leak differently under pressure. Membrane potentials stabilize, but signal propagation stretches in time, forcing neural systems to operate on longer temporal scales. This is why deep-sea animals are not slow by accident. Their biochemistry has been synchronized to compressed motion, where speed is the most limited resource. But compensation has boundaries. At sufficient pressure, water itself reorganizes. Hydrogen bond networks stiffen. Solvent behavior shifts. Protein interiors lose dynamic flexibility, even when stabilized. Beyond this threshold, reactions no longer fail catastrophically. They fade. Rates approach zero. Metabolism becomes mathematically impossible, not biologically fragile. Life does not end because structures collapse. It ends because time runs out. And this defines the true frontier. Not temperature, not chemistry, but kinetics under compression. Where reactions cannot proceed fast enough to sustain order, no adaptation remains available. The deep ocean does not merely test survival. It exposes the precise point where physics closes the door.
After pressure reshapes structure, and after time itself slows at the molecular scale, one constraint remains unavoidable: energy flow. Life is not defined by molecules. It is defined by sustained imbalance, the continuous dissipation of energy to preserve order. Cells respond by minimizing loss. Metabolic networks become sparse. Reactions are chained to reduce dissipation. Heat production drops toward theoretical minimums. This is not optimization for growth. It is optimization for persistence. But there is a final wall. At extreme compression, entropy production required to maintain organization exceeds the energy that can be extracted from the environment. No catalyst can fix this. No molecular architecture can bypass it. At that point, life does not fail violently. It equilibrates. Gradients dissolve. Information degrades. Chemical memory disappears. This boundary is universal. It does not depend on DNA. It does not depend on carbon. It does not depend on Earth. Any system that calls itself alive must continuously export entropy faster than its environment collapses its gradients. When pressure prevents that export, complexity cannot be maintained. This is why the deepest ocean matters. Not because life survives there, but because it shows exactly how far survival can be pushed before physics says no. The abyss is not a curiosity. It is a calibration point, a place where the maximum depth of life is measured, not in meters, but in joules, seconds, and lost degrees of freedom.